Hey guys, I was really sorry. I was trying to go live on Instagram, but my connection isn't strong enough, so I'm still trying to figure it out. I don't understand what the problem is, but okay, so I'm gonna go over the practice questions that I sent you, and I'll try to go over them as quickly as possible so don't take up too much of your time. We're gonna start with these. Don't make fun of me because I had to print these out in red because I ran out of black ink. But it's gonna be all right. So let's go over 1 through 4 first, out of the second packet that I sent you. All right, let's go over those first. So if you can please locate these. I think on the first page is this. This is the Universal Genetic Code chart that uses mRNA and amino acids. So mRNA, that's what we're looking for. That is what is being represented by all of these letters. These are called codons of mRNA, codons, and then the amino acids that they code for are represented by these: serine, proline, threonine, alanine. You don't have to memorize any of that. You just have to know how to use this chart. It's pretty easy because the first base is on this side, second base is on this side, third base is on this side. It's like the wheel, just like the wheel, just a little different. Okay? All right, let's go. So, first question says, using the genetic code chart, fill in the missing amino acids in the amino acid sequence, species A, at the chart above. Okay, so here's for 5 different species, species A, B, C, D, and E. Okay, and there's some missing information here. First of all, species A, this is a section of DNA in species A, okay? This is a section of DNA in species B. This is a section of DNA in species C, and so on. So let's go. First, we have to find the missing amino acids for species A. Okay, that's pretty simple. Let's go to the chart. Okay, so we've got AUG. Since this says mRNA, pretty much all of these are mRNA charts, so they're not DNA charts. You know they're not DNA because look, there's uracils and there's no uracil in DNA. There's only uracil in RNA. There's uracil in all 3 types of RNA: mRNA, tRNA, rRNA. Okay, so let's take a look. First one's AUG. AUG is actually also called the start codon. So if we find AUG, if we go A, first base is on this side. So here's the A region. Then we go to the U, second base, U. So we know where we take the U and the A, we know where in this quadrant. AUC, no. AUU, no. AUG, yes. It's called methionine. It's also called the start codon. That's where an enzyme is going to attach, or a promoter is going to attach and start to read the code. So these are interchangeable. Okay, so we're going to put MET on the line. AUG, MET. Great. MET, you just put the 3 letters of the amino acid. Okay. Next one, GCU, messenger RNA, messenger RNA. We're going GCU, so we're going to go to G, first base, C. C's are in this column, so we're in this box. And it just so happens that all of these, GCU, GCC, GCA, and GCG, all code for alanine. So that's kind of interesting. So let's say there was a little mutation and one of these was switched for one of these, this third base here. That would have no change in the organism because they're still going to produce that same protein in that same spot, that same amino acid, I mean. So if there was a slight base mutation here, that would have no effect on the organism at all. So some mutations have no effect at all, where other mutations have a huge effect. Okay, so this one's going to be alanine. All right, and then if we continue down the line, we're going to end up with glycine, and we're going to end up with serine, and that's 1 point for that question. Okay, next question is, use the information given, fill in the missing mRNA bases in the mRNA strand for species B in the chart. All right, let's go to the chart. Species B. So we've got the DNA, we've got the amino acid, but we don't have the mRNA. Okay, so we have to remember the rule. Adenine bonds to thymine if you're making DNA. But the thing is, there's no thymine in RNA. Instead, we use uracil. So A bonds to U. However, if we see a T, because it's DNA, then T is going to bond to A. If we see a C, it's going to bond to a G. If we see a G, we're going to write C. Awesome. Okay. So TAC, what bonds with T? A. What bonds with A? U. What bonds with C? G. Which makes sense because AUG is always the start codon. See? AUG, AUG, AUG. Okay. TTT, AAA. There's A's in mRNA, so that's fine. GCA, C, G. There's no T's in RNA. U. GGA, C, C. There's no T's, so we're going to put a U. Okay? Okay, so let's go to species C. What is the DNA strand if the mRNA strand is this? Well, we just go back here. Except this time, we are going to say same situation, except now we're making DNA, so there is thymine and there's no uracil. Okay, so uracil and thymine behave similarly. So we see an A, so we're going to put a T. We see a U, which is like a T, so we're going to put an A. We see a G, we're going to put a C. U, 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 those are kind of like T's, right? They bond to adenine, so we're going to put A, A, A. We see a U, so it's kind of like a T, so it bonds to A. C, this one, A. G, G, G. Okay? That's 3 points for that question. Then the last question here says, according to the information, which two species are most closely related? So now we're kind of talking about finding out who's related to who. That's why they do DNA analysis, to find out relationships, like who is the father, right? Or you could just be wanting to find out who's the most closely related to an endangered species of plant or something like that, all right? So if we compare and we go down the line here, what we're going to find out is it's best to compare the amino acids first. Okay, so we see this is methionine, 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 methionine. Okay, because those are the start codons, so they're all going to be the same. Next, we've got alanine, lysine, phenylalanine, histidine, lysine. So these two are the same, right? Let's see. No. Let's see. Okay, glycine, arginine, cysteine, arginine. So we've got an arginine here, we've got a methionine here, we've got a methionine here, and an arginine here. We've got a lysine here, and a lysine here. So far, those two are the most similar. Serine, proline, proline. All right, so there's a similarity between these two here. Arginine, proline. We've got another similarity here, okay? B and E, these two species are going to be the most similar in this gene, okay? Because they have identical amino acids in the same order, which means they're going to make the same protein, okay? So these two are going to be considered the most closely related organisms, species B and species E. So that's the answer for that one, okay? Species B and species E, and you would say that they share I would say something like that. Okay? Let's go to number 5. Okay. Which statement indicates one difference between the gene section of DNA that codes for insulin and the gene that codes for testosterone? Okay, there's no information given about that, so this has to be some general answer that we know is different about two genes that are in different places in the body. So this gene codes for one protein, and this gene codes for a different protein. That's what we know. This one codes for testosterone, this one codes for insulin. Two different types of hormones, right? So 2 different types of genes. So what do we know is a difference between them? Not a similarity. We want to know a difference between those genes. All right. The gene for insulin is replicated in vacuoles. Why would that happen? DNA is not copied in vacuoles. It makes no sense. That's out. The gene for insulin has a different sequence of bases than the gene for testosterone. All right, that one sounds promising. Let's keep going. The gene for insulin is turned on in liver cells, but the gene for testosterone is not. That sounds good. The only problem is that insulin is not produced by liver cells. Okay? Insulin is produced by the pancreas. So if this said the gene for insulin is turned on in the pancreas, but the gene for testosterone is not, that would be great. But this is not true. Okay, this is not true. The liver stores glycogen and turns it into glucose. It interacts with insulin, but insulin isn't produced by the liver cells. So that's out. The gene for insulin is a sequence of 5 different molecular bases, while the gene for testosterone is a sequence of, how would we even know that? There's no way we would know that. Okay, so going back to B, the gene for insulin has a different sequence of molecular bases. That's what makes genes different, is the order of the GATC. That's what they're talking about here. The sequence of bases. The bases are A's, T's, C's, and G's. So it's the sequence of those that makes one gene different from another. That's what it is, okay? Let's go to 6. To determine the identity of their biological parents, adopted children sometimes request DNA tests. These tests involve comparing DNA samples from the child to DNA samples taken from the likely parents. Possible relationships may be determined from these tests because the base sequence of the father determines the base sequence of the offspring. Then it would be a clone of the father. Every kid would be a clone of the father. That's not true. DNA of the parents and the offspring is more similar. They're not saying it's exact. They're saying it's more similar than the DNA of non-family members. Yes, that's definitely true. Okay. Position of the genes on the chromosome is unique to each family member. No, it's not. Because if you look at a chromosome number, let's say this is number 1 from the mom, and this is number 1 from the dad, the genes, the position of the genes are identical. They're just not the same version of that gene, right? So the position of the genes are identical. It's just this person may have a different version or allele for that gene, but the position of them is still going to be identical. Okay? Positions of genes are identical on chromosomes. Like chromosome number 1 has all the same positions. All right? Individuals, mutation rate is the same. No, mutation rate is completely unpredictable, so that's out. All right, let's keep going because my battery is running low now because of all the shenanigans I was doing. Okay. Let's go to number 7.